some information about the sporopollenin. Nothing but what actually the sporopollenin, why, where you are going to see that particular terminology and what is the function of that sporopollenin and the detailed information about the sporopollenin we will discuss in the today's class. And dear students, and we already know that the pollen grains, the mature pollen grain contains the two protective layers. One we call it as the exine, another one is the intine. So can you tell me what actually the pollen grain contains the two layers? One is a protective layer. Now hengi epidermis hengi ಹೇಳ್ತೀವೋ, like we have the walls ಯಾವ ತರ ಇರ್ತದೋ, similarly even the pollen matured pollen grain also contains the two protective layers. One we call it as exine, another one is a intine. So exine, which is outermost one, which is a white quite resistant, quite strong, quite rigid, quite and even unbreakable. And then next to that, we are going to see the intine, and it is intine, which is the innermost layer of the pollen, and which is going to protect again. It is acting also as a protective cell, the protective layer of the pollen, where usually intine, which is quite thinner and which is also not thinner, which is quite easy to break down, where the exine, which is made up of different types of chemical compounds, in that the sporopollenin, which is having a major composition on the exine. So this sporopollenin, because of its rigid, tough, unbreakable properties, the sporopollenin can also be said as the most important, a significant factor in studying the pollen grains and their the reverse. I mean the carbon dating and curricula. I mean, the reverse study of from the pollen grain. We are going to study how, if the pollen grain is like this, then what would be the how would be the tree, and when it was that particular plant was available. And we can study even the eras also. I mean the Carboniferous era, even Jurassic era, even the different types of era, Mesozoic era, all those we can study. And that is again with regard to the carbon date. So you all can able to see this one, the A and B image. You can see that this particular one is an intine one, which is quite smoother and smooth. Nothing but we are not going to see such articulation, structural what you call articulation anywhere in the intine region. So on the pollen grain, so if you cut it, if you cut in a small portion, like just like a cake, you are going to get like this. So this one is the intine, which is the innermost one, and then next to this, we are going to see the what we call the vegetative cell, as well as the another one is the reproductive cell. That is a male, as the two male gametes. And this one is a blue color one. We are going to see such variation. I mean the structural modification we are going to see. So this one is exine, another one the exine region. Could you all find the difference in the A image as well as the B image? There you can see the exine containing this sporopollenin quite different. The accumulation of a sporopollenin on the exine is quite different. Just see the differences in the structure of the outermost layer in a pollen. This looks like a single dumbbell shape, and here you're going to see a much thickened one with a small cavities over on there. Right? So such variation in the articulations of a in the exine is mainly due to the sporopollenin. Fine. And apart from that, we are going to see some the fatty substances like hydrophobic substances are also going to be released by the tapetum. Nothing but the exine, which is the major components of an exine, are just because of a secretion made by the tapetum itself. As and when there is an development in the microspore formation, we are going to see the tapetum is going to lose its presence, lose its existence, and meanwhile, subsequently, it is going to release some of the substances, because the tapetum is mainly having the role of nourishing the developing microspore. Apart from that, as they complete their what we call the development stage, as when they are on the microspore at the mature stage, they are going to release some of the molecules such as enzymes, scalase enzymes, and that particular enzyme which will make the pollen tetrad into a haploid single individual pollen. And along with that, it is going to release some of the substances, and which contains the precursor for the sporopollenin. That's what we call it as a pro-Eugeist bodies. And apart from the pro-Eugeist bodies are going to be released. Some of the precursors for the formation of a sporopollenin are going to be released. All the things are going to be released directly from the tapetum. Apart from that, they are also going to release some of the hydrophobic substances. Nothing but they are quite fatty, substances. They may be unsaturated fatty substances or saturated fatty substances. They are also going to be released by that. And now you may ask why the tapetum is going to release, or why this particular anther needs to have the sporopollenin as well as the fatty substances as well as the intine. Usually intine contains the cellulose, hemicellulose, and as well as the callus, and we'll talk about the intine and exine when we talk about the pollen structure. Fine. And before that, today we'll stick ourselves to this sporopollenin. As I already told you that this sporopollenin, which is a major component, major composition in the exine of a pollen grain. Let me tell you again, make more precise that the mature pollen grain, a mature haploid individual pollen grain will be having the sporopollenin content in the exine. Apart from that, they also contain the fatty substances. Why? Because these pollen grains are, if they get moistened, there is a possibility of the what we call pathogen accumulation of the pollen grain. Then they may lose their activity. So to avoid that one, the intelligence, I mean the pollen grains will be having some outer depositions on the exine, on the sporopollen content to have these fatty substances. What happens when the pollen grain, though they just like looks like a dust particle or a dust like substances, because of the fatty molecules which are available, which are on the pollen grain, they won't get affected even after the moisture content. Only when they found a suitable compatible stigma, then they are going to lose their all the chemical compositions, and finally we are going to see the formation or a development of a pollen tube, and that is what you are going to study in the fertilization. So coming to the sporopollenin, and before talking about in detailed chemical composition and the structure of a sporopollenin, let me give you some information, some introduction or remarks on the sporopollenin. Again, yes, I already told you that it is again considered as outermost layer of exine. Okay, na? So this sporopollenin can also be said as a cloth or a fabric of exine, and which is extraordinarily a resistant material. So now you may ask why it is been said as extraordinarily resistant material, because this particular sporopollenin, which is even till today, 20 July 2021, we are not yet revealed the actual chemical composition of the sporopollenin. Because of that, why? Because we are not yet found any suitable chemical agent which can break the sporopollenin. We are not yet identified any specialized enzyme which can break the sporopollenin, and we are not yet identified any strong acid or a strong base, generally strong chemical agent or a reagent which can have the which can have the capacity to break down this sporopollenin content. Because though it is an organic one, because the plants which are on their own naturally they are going to release some substances, and they are making this sporopollenin which is going to be attached on the exon. Nothing is artificial here, though it is organic one, but it is quite resistant enough. It is a highly significantly resistant one because of non-availability of any agent which can have the capacity to break down the sporopollenin content which is available in the exon. Now you may ask, then when it is not being broke down, when it is not being studied without having any of the such chemicals which have the capacity to simplify the complex structure of a sporopollenin, then how come we can know that this particular sporopollenin contains the different types of the chemicals? Again, how to elucidate the structure of a sporopollenin? So in view of that, many of the scientific community come forward to study the composition, chemical composition and the structure, chemical structure, and because of the non-availability of the compound which is acting as an anti-sporopollenin, nothing but which a agent we need to have so that this particular complex sporopollenin should get broken down, should get easy to study what are the composition, what are the chemical structure, what is the chemical, what are the chemical composition available in the sporopollenin. Because of non-availability of that, so we are till now we are not at complete this particular sporopollenin which is quite resistant. Why? Because it is even till today we are not at identified any suitable compound which can break this sporopollenin content. Why? Because any of the complex structure needs to be broke down. Then nothing but it has to get simplified. Then only we can study the each and every what we call information about the chemical characteristics as well as the chemical structure of a compound. Because of non-availability of that, we are not yet clearly understood what are the components, how this sporopollenin which is made up of, and what are the chemicals which are making that particular sporopollenin into one more property. I mean, let me talk about one more important information about this sporopollenin is that this particular sporopollenin is quite stable in such a way that even if you heat at a 300 degrees Celsius, in some of the articles they have given even it has a 400 degrees Celsius also. Even if you heat the pollen grain at a 400 degrees Celsius, this particular sporopollen is not going to lose its capacity, its stability. Such high temperature controls that they are highly thermophilic in nature. Yeah, that's what.
Sporopollenin.
Yeah, because of that. ಈಗ ನಾನು ಈಗ ಅದೇ ಹೇಳ್ತಿದ್ದೇನೆ. So because of this particular tough characteristics, the most of the pollen grains, the imprints of a pollen grains or the fossil evidences of the pollen grains are we can see in the sedimentary rocks. Nothing but the
Yes, sir.
even ನಾನು ಬರ್ದಿದ್ದೇನೆ 500 million years old fossil evidences or we have got, and through those 500 million year old fossil evidences of the pollen grain is just because of the sporopollenin content, because the scientific community have already taken those the fossil evidences and they have gone a detailed study of those fossils and they came to know that this contains a tough chemical compound even after the 500 million years are old, or even after the 500 million years also it has not lost its existence, it has not lost its the structure, it has not lost its chemical characteristic. Just see, after 500 million years, I'm talking about 500 million years, not just 500 years. ನಾನು ಇರ್ಬೋದು 500 years, ಐ ಡೋಂಟ್ ನೋ ನೀವು ಇರ್ತಿರಲ್ಲ ಗೊತ್ತಿಲ್ಲ ನನಗೆ. ನೀವು 500 million years ಅಂದ್ರೆ so just see how tough it is, and 500 million years ಆಗ್ಬೇಕಂದ್ರೆ just see what is the evolutionary characteristics it has taken. Such a big gigantic dinosaurs have lost their existence. A simple microscopic pollen grain which containing still minute, still nano, still picogram level the sporopollen existing as it is without losing its the what we call the original characteristic. So because of this, the chemical structure, chemical composition of a sporopollenin is inadequately known. Let me tell you guys, last year when I was talking about this particular class, I told most of the students that it is not clearly known and studied. But in the recent years, in the year 2019, 20,
Yes, sir.
21, we have got some of the information about the sporopollenin. Because of that, I have changed the word it as inadequately known. Okay, now we'll go to the next one. Now we'll talk about the chemical composition of a sporopollenin, and it has been said that most because the when we nothing but just completely they are the sporopollenin, we already know that the precursors for the sporopollenin are going to be released by the tapetum, right? So the tapetum contains what are the compositions the tapetum, come to know what are the possible depositions we are going to see it on the exam. Similarly, when the scientific community have started to study, they came to know that the sporopollenin is made up of assumed that not it has been clearly elucidated. Fine, it has been assumed that the chief component of a sporopollenin is the polymer. Many of the monomers are cross-linked, not they are directly linked to each other, they are not laterally linked to each other, they are cross-linked, making into a complex structure, and there are those cross-linking is rendered by the ether linkages, and they also contains and the usually any of the atom so any of the chemical when you start when we talk about the chemical structure, we are going to see the three backbones. So that is made up of a carbon, hydrogen, oxygen. The components of a sporopollenin, the many of the scientific community partially or at a close level they have made they have studied the composition of the sporopollen, and finally they have got that result as they contains the carbon, hydrogen, and oxygen, and all these together the carbon, hydrogen, oxygen meioty present in the sporopollenin along with the polymer, that particular makes a complex polymer structure. And it has been because it has been synthesized naturally by the tapetum as and when there was a development in the in the anther wall development as well as a microspore development. When the microspore reaches the maturation stage, that particular secretion released by the tapetum are going to be uh, deposited on the this particular pollen grain. So because of that, we call it as a biopollen. And this particular biopolymer not only contains the what we call the cross linkage of a monomers with ether bondings. They also contains the long chain of fatty acids, that is phenylpropanoids. Along with that, they also contains the phenolic substances and the traces of the xanthophyll as well as the carotenoids. This particular sporopollenin structure, apart from containing the complex the polymers, they also contain long chain fatty acids, phenylpropanoids, phenolics, as well as the traces. Not the, much of the carotenoids and xanthophylls. They contain the traces of carotenoids as well as the xanthophylls. And many of the some of the experiments have also shown that they also contains the major precursor, that is a the precursor for the phenylalanine. What is that? These particular sporopollenin contains some of the precursors for the phenylalanine as well as the some of the carbon required. Carbon source thing, but any of the mechanism requires energy source, and that particular energy source is mainly rendered by the carbon. That particular carbon may be in the form of a carbohydrate. So these precursors for the phenylalanine as well as the precursor for the carbon source are also available in the sporopollenin. Some of the experiments have proven that. Apart from that, they also contain some of the aromatic structures like para-coumarate as well as niranjanin. So these two are also some of the compounds. Let me tell you more precisely that all the sporopollenin doesn't contains all the chemical compounds what I'm explaining you to you. Okay, and some of the sporopollenin derived from some angiospermic plants, some of the monocotyledon plants, some of the dicotyledon plants, some of the pteridophytic plants. I mean, in the cone-bearing plants, and some of the this one the gymnospermic plant. The composition of a sporopollenin is quite different from plant to plant. Even when it comes to the flowering plant like the angiosperms, they also the composition of a sporopollenin varies. So, and in general, I'm just giving the picture about the sporopollenin that some of the aromatic structures like the para-coumarate as well as naringenin is also a compound which is going to be seen, which has been observed from the sporopollenin, and that is what the tapetal cells, as I already told you, that the main composition precursors for needed for the sporopollenin are released by the tapetum system as they are reaching as the microspores are at the stage of a mature stage. They are going to release the lipophilic globules, and these lipophilic globules, which are considered as the precursors for the synthesis or a formation of a sporopollenin, and where these will get accumulated with the pro-ubist bodies and become the ubist bodies, and they are going to be settled on the exile. And the main structure of a sporopollenin is that they contain simple aliphatic polymers associated with aromatic conjugated side chains. So in generally, the main structure revealed by some of the scientific community, and it has been accepted by most of the biologists as well as the phenologists, that simple aliphatic polymers associated with aromatic conjugated side chains. And now coming to the what are the variations DNA? Now we'll talk about the chemical variation in the different plants. Different plants, andre, it may be in angiosperms, between the angiosperm and the gymnosperms, between the cones of different plants. Cone-bearing plants and pteridophytes barbodu, even the one gymnosperms. And it has been, as I already told you, that when the sporopollenin moiety has been taken for the understanding the backbone, it has been finally revealed that it contains the carbon, hydrogen, and oxygen molecules, where the NNMR, nitrogen nuclear magnetic resonance tool, has revealed that this sporopollenin doesn't contain any of the nitrogen molecules. Nitrogen atoms. And it has been said that very recently in the year 2019, we have got an article that the composition of the carbon and hydrogen, I think I have not put that particular information here in the slide. If you want to just write it, the composition of a carbon and hydrogen always in a 5:8 ratio. Nothing but 5 molecules of carbon ಇದೆ ಅಂದ್ರೆ 8 molecules of hydrogen ಇರ್ತದೆ. It has been common in all the plants of a pollen grain containing the sporopollen. Their number may be vary. ಒಂದು 50 ಇದ್ರೆ ಇಲ್ಲಿ 90 ಇರ್ತದೆ, ಒಂದರಲ್ಲಿ 5 ಇದ್ರೆ ಇದರಲ್ಲಿ 8 ಇರ್ತದೆ. So the number of the carbon molecules, because it's a polymer, right? The number of the carbon as well as hydrogen molecules may vary from the plant to plant, pollen to pollen, the sporopollenin content to sporopollenin content, but it has been generally said that 5 raised to 2 or 5 raised to 8 is the composition ratio of the carbon and hydrogen. Apart from that, in some of the organisms we have some of the even the author's name also in the present slide. The in the Lycopodium, as per the opinion of Just as well as the Visari, in the year 1931 have said that the Lycopodium pollen, I mean the male microspore contains the sporopollenin having 90 carbon atoms, 142 hydrogen atoms, and the 27 oxygen atoms. And apart from when this what the carbon, hydrogen as well as oxygen molecule in number, and when it comes to the structure of the actual chemical composition of the microspores in the Lycopodium or the macrocyclic backbone. Nothing but this again macro, it's a again which is a quite strong, which is a quite bigger one, and they are contains the polyhydroxylated tetraketide like monomers. Nothing but hydroxylated tetraketide monomers are making it a big n number of polymers, along with that false aromatic 2 pyrrone rings. In the 1931, these two persons have revealed the composition of the microspore containing sporopollenin in the Lycopodium. And let me make one second. So now you may ask why this particular in the research methodology. When I'm talking about the review of literature, we have to write the author's name in a bracket. So this particular information I have got it from some article, and just see, this is what the opinion of these two persons. You have to write like this. Okay, sir. And this is not my original one. This has been drawn from this research article, which has been said by Just as and Vicari in the year 1930. Now, coming back to the embryology, and in the Typha angustifolia also, the two persons independently, Hess and Athels, these two persons are individually in a different years in the 1995 and 2000 respectively, has given the presence or occurrence of the two important components on the sporopollenin of a pollen grain of a Typha angustifolia, and this one is a benzoic acid meiotis, presence of benzoic acid meiotis on the sporopollenin of a Typha angustifolia by Hesse, and alpha beta unsaturated carbonyl group by Els in the year 2000 in the present in the sporopollenin of a Typha angustifolia. In my previous, the next person, Wang and others, et al. So what actually mean of et al. And others, more than 2 authors included, and have done research on the sporopollenin of a pinus microspores, and they came to know that presence of the occurrence of a polyvinyl alcohol units. Along with that, they also contain aliphatic monomers, which are cross-linked with a series of the acetal linkages in the pinus microspores of the sporopollenin. And apart from that, even in some of the articles have also shown that the presence of a microspores in the algae, especially in the green algae, also contain the sporopollenin, and they we have the Phycopeltis as well as the chlorella. These two are spores. They also shown the presence or the occurrence of a sporopollenin. These two also tendered by the Good and Chapman in the year 1978 and the Atkinson in the year 1972 respectively. And this is what about the chemical variation. We are going to see it in the sporopollenin. And now we are at the last bit of the today's class. What are the properties? Why we are supposed to make sure, why they are important, what are the different properties of sporopollenin here? Because of that particular tough characteristic, unbreakable characteristics, you know that pollen grain contain the DNA. You know that the DNA, when it get exposed to some of the mutagenic agents, let us take, we have the UV is a mutagenic agent, radiation, and they may alter the sequences of the DNA. So this particular sporopollenin because of this particular stable, unchangeable, unbreakable, resistant characteristic. It is going to protect the internal DNA of the pollen grain, right? Then second property of the sporopollenin is that it is resistant to acetylysis. Nothing but if you even use some of the acetic components to break down this sporopollenin, it is not going to change its characteristic or a stable characteristic. And because they are resistant even to high temperature, as I already told you that they have found stable even at a 300 degrees Celsius, and we have not yet any identified individual or a complex chemical which can break the sporopollenin, and we have not yet received any of the physical as well as a biological agent which may be released, which may be synthesized by any of the microorganism or a fungal or a human or a plant or any of the organism which can break the what we call the sporopollenin. This is what are important characteristics. And one more point, one more property is because this is a tough compound. Because of that, the tough characteristics we are even till today we are having the fossil evidences of the sporopollenin, and because of that we can even study the carbon dating. Carbon of the pollen in such a way that we are going to see the sculpture. Nothing but ನಿಮಗೆ ಎಲ್ಲಿದೆ ಅಂದ್ರೆ ವಿಲ್ ಶೋ ಯು ಟು ದಿ ಪ್ರೀವಿಯಸ್ ಸ್ಲೈಡ್ ಅಲ್ಲಿ ಹೋಗ್ತೇನೆ. ಎಲ್ಲಿದೆ? ಓಕೆ. ಸೊ ದಿಸ್ ನಾನು ನಿಮ್ಗೆ ಆದ್ಮೇಲೆ ತೋರಿಸ್ತೇನೆ. Because of some ornamental characteristics of the exine, different beautiful how the girls and women ಆಭರಣ ಹೆಂಗೆ ಹಾಕೊಳ್ತಾರಲ್ಲ, similarly even the diatoms ಹೆಂಗೆ different diatom structure ಅಂತ, similarly the pollen grain structure, the sculptured characteristic of a pollen is just because of the sporopollenin. And this is what very recently we have got this sporopollenin structure. This is what like this biopolymer containing carbon, hydrogen as well as the oxygen molecules. This is what a structure. And we'll show you one image perspective here. First, we'll end this session. So then, can you all able to see the screen?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Can you all able to see this? These are the pollen grains. Just see how the articulation, how the sculpturing, because of this sporopollenin. So if you go and scroll, we are going to see the most interesting pollen grain. Just see how it looks.